Okay, welcome everyone to today's North Central IPM Center Pests in Progress series. Today's webinar is Are You Smarter Than a Lone Star Tick? My name is Laura Isles. I'm one of the co-directors of the North Central IPM Center. We're one of 4 regional IPM centers supported by USDA NIFA Crop Protection and Pest Management Regional Coordination Program. We all have a mission to coordinate IPM across the region by sharing and promoting IPM research extension and teaching activities. So during today's webinar, if you have questions at any time during the webinar, please type them in the Q&A portion at the bottom of your Zoom screen. And after our speaker is done, we'll go through and answer all those questions. So again, usually at the bottom of your Zoom screen, you should see something that says Q&A where you can type in your questions. So I'm excited to introduce Holly Graff from Old Dominion University to talk to us about Lone Star ticks. So Holly, I'll let you take it away.
Awesome. Thank you so much, Laura, and thank you for the invitation to talk to everyone today about Lone Star ticks and all the fun things that we've learned in my lab here at ODU over the years about Lone Stars in general. And so anytime I talk about ticks, I think it's good to take a little bit of a step back and make sure we all understand where ticks are and where they fit into the world. There's only about 875 species of them worldwide. We have somewhere around 15 or so here in the state of Virginia. There's around the world, they're everywhere. They're on all continents, including Antarctica, so there really is nowhere you can get away from them. Um, Interestingly, they're not technically insects, right? They are acarids, and I think that sometimes comes into play when we're looking at understanding their life history or possibly interactions with insecticides or a few other things. And so keeping that in mind is always a good little biological fact that could play into understanding how their biology works. And of course, they're vectors of human pathogens, which we know here in the US, Virginia Hampton Roads, they are the #1 vector of human pathogens. Most specifically, Lyme disease is the most common, but we'll talk about some other things today. And second in the world to mosquitoes for vectors of human pathogens. They, of course, also vector veterinarian and wildlife pathogens. They can be incredibly host specific in some life stages. They can be very broad in their life stages. There's a lot of variation about a lot of things about ticks, and one of the hardest things about studying them is there's difficult to generalize from one species to another, or even from one life stage to another, to understand how all of these things work. But they pretty much feed on every vertebrate animal on the planet has some tick that will feed on it. So there is nowhere you can go to be safe, and there's nothing that is safe on the planet. So it is kind of amazing how widespread they are in their history. From their life cycle, I think one of the things again here is important to understand is it's a longer life cycle than a lot of other vectors. And so when I often think about vectors, we think about mosquitoes, they're very short-lived. Ticks, on the other hand, have this punctuated life history, and so they have 3 blood requiring life stages. So larvae, and if they're successful in feeding, they'll become nymphs. If they are successful in feeding, they become adults. The adults lay eggs, and the cycle repeats. There are soft ticks, which have a little bit of a different life history, but I'm going to still focus on the hard ticks today, these exotid ticks. They typically take 1 blood meal per life stage. That's their goal, is to get a full blood meal. It can last 3 to 7 to 14 days or longer, depending on where they're what animal they're on and which life stage they are. And that female can lay between 500 and 5,000 eggs and kick this whole process off again. We classify ticks as 1, 2, or 3 hosts, depending on how they use their hosts. And so there are ticks that after larvae, when they climb onto that animal, they will just remain in the fur of the animal. But the majority of the ticks, and the one I'm going to talk about today, which is Lone Stars, are 3 host ticks. So between each life stage, they fall off and they live in that leaf litter or brush for months to years between feeding. And so thinking about understanding the dynamics of when and where and why you find ticks really has to take into account these long periods of time between interactions with different kinds of hosts potentially in each life stage. So again, today I'm going to look at lone stars, and just as a comment here, the lone stars do not feed on small mammals or birds very often. You will find them sometimes, but their true preference is going to be large animals, maybe some medium-sized mammals we'll find them on as well. But really, a lone star's personal favorite food is white-tailed deer. And so, unlike black-legged ticks, which is what people tend to more understand and has been out in the literature a lot longer. Larvae don't feed on small mammals, larvae will feed on us. And so all three life stages are looking for something large coming through. And so, of course, the question often comes up of why are we studying ticks now? What's the big difference? And so, tick-borne diseases, I got into working on ticks during grad school in the mid 90s. And so, in the mid 90s, the map down here on the bottom left is the range of Lone Star ticks. And it had just moved through Tennessee into Kentucky at that time, and that was the kind of the northern range of the ticks. And there was an outbreak of ehrlichiosis in Cumberland County, Tennessee, and I got interested in this and started looking at it mostly at that point from a mathematical risk assessment modeling idea and really trying to understand when and where and why we find ticks. And these outbreaks seem to be come and go. They don't seem to have a lot of prediction. Lyme disease was just coming onto the scene. But again, I was really interested in Lone Stars because of where I lived and what was going on around me. And those range expansions are really, really fast, right? So 20 some years later, right? That northern edge of Lone Stars has now moved all the way up halfway through Michigan. So this map on the right is the estimated distribution is in yellow in 2024 of where Lone Star ticks have gone. So they've moved through the entire states of Indiana, Ohio, Illinois, all the way up into the Dakotas now, up into Maine. And so they're just everywhere, and this expansion is very fast, probably more related to land use than climate change based on the speed, but they are now everywhere. And so I think it's important for all of us to truly understand the differences of lone stars compared to black-legged ticks, because there is quite a bit of difference in understanding them and hopefully not feeding them by at least you, right? So here at Old Dominion University, I'm going to base a lot of the things out of the stuff that we've done over the years here. So we have been running a long-term study of tick ecology and tick-borne diseases in Hampton Roads, southeastern Virginia. So I had mentioned I had started working on ticks and tick-borne diseases in graduate school in the 90s and had been working on developing mathematical models and understanding what was going on, and really struggled for 20 years, the first 20 years, to build a good model. And the challenge really was that I could not find any decent long-term data set thinking about modeling. And I was after going through many different experts and different people that had been in the field for a long period of time, became convinced by all of them that no one had really ever collected ticks thinking about modeling, thinking about these long-term data sets of how we do these. And so off I went in 2009, being told it would be very easy and I could come back inside pretty quickly and do math again. And I found out that was not true. So I started collecting ticks from the field very quickly, and then we bring the ticks back to the lab, and so we then test them for pathogens. We look to see what their population genetics look like. We look at a lot of other things, and then eventually we get back to sitting down and doing some modeling and simulation to understand where risks are, what control possibly we could do. So we started off doing this, and so we do mostly flagging, right? So you take your white denim cloth, attach it to a dowel rod, drag it through the vegetation, pick off all the ticks that you can find on that flag, put them in a vial, bring them back to the lab. That, of course, gets any tick that's up questing at the level that would be attracted to a large animal, which is what myself and my students represent. But you don't capture all of the details that are going on because not everything will be attaching to these flags. If they're interested in rodents or other things, they'll be at different levels and different attractants. They may just even be there and ignore you. So we also have done rodent live trapping. We've gone to hunt check stations. We stop at roadkill, which is always a really sexy opportunity. We work with veterinarians. We get ticks off people. We pick ticks off birds. We pick ticks off reptiles. Pretty much anything we can get our hands on to really try and get this picture of what's going on in the Hampton Roads area and kind of use that as an understanding of at least what we know has been changing in Virginia. So today I'm going to look at mostly just the flagging data. So this is data from consistent flagging. And so what we've done is we've gone out here down in Virginia. I'm in southeastern Virginia and Hampton Roads area. And we have some field sites. And so the red dots down here on the map, the blown up map, are regular field sites where we have established transects, and we have sampled those transects every month since 2009. And over the years, we've added other projects. So the blue dots are some projects we added a couple years ago to look at, north-south differences in Virginia. We've got some volunteers and some of the green dots that are starting to fill in some of our data in the last year. But the majority of the data that I'm going to show you today is down in these red long-term sites. And so The advantage we have is by doing the same thing in the same way and the same everything over so many years, we can start to understand what's happening our area for ticks, right? So what does that look like for data? So I omitted 2009 because by the time I figured out how to get permits and how to do all the things, the year was really not fully represented. And so I omit that. But 2010 to 2025, we normalize by effort. And so this is kind of taking into account that, field sites come and go a little bit with some change, which we'll talk about. We also have some differences, and we might get rained out for a month, or there a little changes here and there. So we normalize it by effort just to make sure that we have all of the information. And you can see that there's some trends here, right? So anywhere in any given year, we usually get between 25 and 60,000 ticks that we will bring back, sort by species and life stage, process as many of them as we can afford to do from PCR to understand what pathogens they have, and try to get some dynamics of what's going on. And so if I drill down into this picture here, right, so you can immediately see some data and without even being, all that fancy in your analysis and say, well, look, 2012 was a bumper crop year, and boy, was it ever. That was about 3 years into my study, and I was really regretting my decisions to move into tick collection because there was just infinite ticks that year, it felt like. And here in Hampton Roads, the 2012 actually also happened to be the warmest winter we've ever had on record. And it was a year we didn't get below freezing that winter. We had 90 degrees a couple times in February. And so here I am 3 years into my study, and I was like, oh, I understand now that warm winters equal more ticks. And I'm like, well, not sure if I want to publish that just yet. I better understand these things a little bit more. So I patiently sat on my data, and you fast forward through time and you see 2018 is another bumper crop year. Well, 2018 is actually one of the colder records and winters on record until this past winter. We had a ton of snow, we had a ton of freezing cold weather, and yet we still got the second highest ticks that we've ever collected. And so it kind of made me realize very quickly at the data that I'm looking at that there are two factors into understanding the tick data that you collect when you collect it in this manner. And the first is, yes, it reflects the total density of ticks that are there, but it also reflects the number of hosts that are around that could absorb these ticks. As I tell my grad students and undergrads all the time, we're collecting what we call loser ticks. And so these ticks may be a lot of them in the environment because there are a lot of them in the environment, or may they be a lot of them in the environment because what they would have fed on may not be there anymore, right? So they fed on a host a year ago, a whole year has transpired, and a whole bunch of things have changed underneath that to be able to say, well, maybe now that host is gone. And so all those ticks came back up looking for the deer that was there a year ago. That deer got harvested, the deer got moved on to a different area. And so now all those ticks are there looking for what used to have been there. And so these echoes kind of exist over time. And I think that's what happened in 2020 as well. So 2020, obviously, we had COVID. This was one of the few pieces of research that we could do was to go out and try to collect ticks, and we were all excited to actually do something. So we still went out just equally amount in 2020, but in that year, animals were actually far more active because of the lack of human activity. And so I don't think it was necessarily the quietest tick year we've ever had, but I think it was more a reflection of probably there were a lot more host activity going on because the animals were moving in a very different manner. And so as I look at these data, I have to try and think about all these complex things that are going on. Now, I told you I was talking about the Lone Star tick today, right? So let's drill into that. And so those dynamics that I just showed you were all ticks collected, but the Lone Star tick, right, named for the dot on the back of the adult female, will feed on humans on all life stages, likes those large animals. And if I just filter that data to only Lone Stars, it looks almost identical to what I just talked you through. So here are some stacked bar graphs where the big blue line are the larvae that we collect in a year, the orange are the nymphs, and the grays are the adults. And so again, this is normalized data over the years, and you can see that it still goes almost to 60,000 because again, 95% of the ticks that we collect in any given year are Lone Star ticks. That is what we see all the time. And that's what follows these trends of, warm winters seem to give us a lot, cold winters seem to give us a lot, weird human years seem to give us few. And so trying to really understand, well, maybe that was a low year, right? So we have to try to figure out how do we tease apart all these different layers. And that's where some of the exploration that we did goes into this, as well as some of the modeling comes back into this, right? So we figure out the phenology over time, right? So these I can make these graphs every year. We understand in our area, adult lone stars are up in May, June, right? And then I can see how each year plays to the years before. So here are my orange bars are the average for all of the years we've ever been out everywhere, and blue dots give us the average per year. So I can say, okay, 2025 was a little bit low on the adults. The blue dots here for the nymphs, a little bit low on the nymphs, but we have a whole lot of extra larvae, right? Larvae are a little harder to interpret, but we can certainly look at our trends of larvae over time and get a better idea. Well, maybe larvae this year should give me more nymphs next year, but that's a whole year away, and there's all kinds of things that can happen in the meanwhile. So I need to understand, how do I filter the data from one life stage in one year to predict or understand what the risks are for the next life stage in the next year? And that's one of the pieces of research that we're trying to tease apart to better understand and interpret the information we get from the field. So just to make sure we're all lovely pictures of lone stars, right? All kinds of good times. These are the larvae. We get them in large bunches, right? People often call them seed ticks, but you can also get a bunch of adults, and this is a case of mutiny happening, right? Engorged ticks, still females will still have that dot, but these ticks are just incredible in their aggressiveness, right? So if you can see these videos moving a little bit, this is larvae on the boot of one of my graduate students, and then there's about 10 nymphs crawling up on this boot as well. And so these ticks will crawl up you very quickly, and they'll try to bite you. We do find them in all of our habitats, right? They do really like ecotones in woods, so the edges of woods between edge and grass, but you will still find these guys in tall grass. You can find them in the parking lot. They will go long distances to try and find a host. And the reason we got into it originally, right, I said was ehrlichiosis, right? So just to make sure we're on the same page for understanding that, right? So Ehrlichia chafiensis is a gram-negative intercellular bacteria, right? The reservoir host is white-tailed deer, so you can know where that feeds into all of this. And like every tick-borne disease known to mankind has the same kind of symptoms where you have fever, headache, myalgia. That seems to be the common theme. If you look at the cases of Ehrlichia, right, I got into it in the even before the graph existed. So um, there was a change in case definition, which is why the colors change. But with the new case definition, you can see that other than the 2020 kind of quiet year because of reporting, probably, there's been a pretty significant increase over time in reported cases of ehrlichiosis. If we look at where, we are right in the heart of that, right? And so this is kind of one of the diseases we're chasing to understand how Virginia reflects what's going on. And we've tested our ticks, and we rarely find the pathogen, but when we do, we find it in large numbers. And so it is hyperfocal in the when and where, and you get up to 5% positive or 8% if you're doing Ehrlichia in a particular population of ticks. And so now we're working on trying to pull back apart and understand, how do these positives relate to human cases. And so far, we haven't been able to crack that nut, but we have the pieces, right? And so we're missing some other layers of understanding of how ticks that we find relate to ticks that would bite people. So working on that. Um, just so you're aware, the other big interest for Lone Star ticks in our area is the southern tick-associated rash illness, and so we don't know what pathogen causes it, but it is associated with the bite of a Lone Star tick. The rash resembles Lyme disease. The geographic distribution pretty much matches Lone Star ticks, but it seems we don't know much about this at this point. So there's a lot of work to be done on the biomedical side to understand star eye. But I just like to bring it up anytime I'm talking about Lone Stars, because Lone Star ticks cannot biologically transmit the pathogen that causes true Lyme disease, Borrelia burgdorferi sensu stricto, but it can transmit things that look and act a lot like it. And so it's something to be aware of. The big one, of course, that has everyone's attention now, is I've stood for years in front of people talking about ticks, that you could die, you could have really bad illnesses, you could have all these things. But it get a lot more traction when I start talking to people about how it's going to impact their life, right? So allergies are no joke. Allergic allergies to mammal meat, as well as milk, as well as all the other things associated with alpha-gal, this allergen that's found in tick saliva and a protein in non-primate mammals, and it can have a delayed allergy attack, a delayed anaphylaxis, to both mammal meat as well as other mammal products. And the ubiquity of the animal products in our current consumables is phenomenal in the pharmaceuticals, in foods, and across the board. And if I look at cases, of course, I live in a nice hotspot for this, but anywhere the Lone Stars are, so it pretty much matches the Ehrlichia map. Except there's, of course, the additional outbreak up here in Minnesota and Wisconsin. So there are other ticks other than the Lone Star, but the Lone Star is the majority of the cases in the US, but there are a lot of other ways to have this allergy triggered. And so it's something more recently being studied and to be understood. We didn't really have an understanding of this until about 10 or 15 years ago. So there's a lot of work to be done on this. So all that, now we know that we have ticks, we have pathogens, they cause diseases. So we want to start teasing apart some of those connections between different life stages. And so some of the question becomes that macro level often misses micro level changes. And so local interesting changes we've looked at, I showed you these data. I was like, look, this year was lots of ticks, this year was low ticks. But if I drill down and actually look at some of the field sites themselves, I get some different answers, right? So this was the data that we had seen earlier. And so again, 2012 was high, 2018 was high, 2020 and 2024 were low, right? I drill in, let's go into the site that's pretty much on the ocean, right? So we're right on the beach. It's a beautiful field site. Everybody likes to go there, right? And they're going to be able to ask lots of cool questions. And if you look at the data that we've collected out there right on the beach, we don't see that similar pattern, right? We see this massive drop off up until about 2023. And so what we're actually watching here, if you want to look at the relationship in this one, and the green bar here is the Lone Star ticks, but all ticks dropped off in this site. And the best biological understanding we have is we actually are starting to have so much sea level rise that this site is inundated with seawater more often than it's not. And so we had a couple major hurricanes in 2012 and in 2016, 15, 2015, 16 era that really overwashed these areas and probably made it uninhabitable for the hosts, which eventually drove out the ticks. Right? If I flash forward over to a site in Portsmouth, right, I get a similar experience, right? I have an unbelievable number of ticks. This site in Portsmouth was a wildlife preserve that gave us more ticks than you could shake a stick at. And then suddenly everything went away. And we went back and looked at the biology of this time, this 2020, 13, 2014. It went from being an ecological nightmare of more deer and raccoons than you could shake and then you could count. And then coyotes moved in, and coyotes moved in and brought those tick food animals, for lack of a better word, back down to better ecological balance. And as they did that, they managed to pull the tick populations down with them. At least that's our best guess and understanding at this point. And so it's interesting how you get these two data sets that look very similar. One is caused by, oceanographic changes, and one is caused by a biological changes. So again, it's important to really understand the systems you're looking at to understand the data you're looking at. Right? And just to make sure you know that I don't just collect all ticks out, because I get asked that a lot. And boy, if I could collect the ticks out of a site by just collecting what we collect, I would have a business, not, be rich at this point. So if I go over to the Eastern Shore, there are plenty of places where I've done the same things and I've had high years, low years, high years back, and then 2025 was back up. So it again, there's no real pattern that I've been able to figure out at this point yet. And so as I look at these data, I have to also think, what level am I looking at? What questions am I looking at? And what's going on? So we get them everywhere in lots of places, and I can answer some questions about some of these dynamics, but there's so much more I don't understand. And so I ended up with my students over the years. We've done a lot of field and lab studies to both look for weaknesses, right? Because people hate ticks and they want to get rid of all of them. And so maybe if we could figure out what drives those populations down in those years and understand maybe it's something we could help with, or at least control it in some local areas where we want to have better tick control, and then leave the ticks to have their own life where we don't go. And so we started doing a bunch of different things as students asked questions over the years. A lot of these were derived from things that students would ask while you're driving to field sites and you're collecting hundreds of ticks. You think a lot about what you're looking at. And so for most of the tests we're going to look at, I'm going to talk about what's called a circle test. So I wanted to just show you, if you've never seen that before, anytime you're analyzing ticks for dead or alive, we draw a circle on a piece of paper inside of a double-sided sticky tape, put the tick on its back or its front there, and then blow on them gently and see if they can right themselves and get themselves out of their circle, in which case we declare them to be alive. So if I say the circle test or dead or alive, that we actually have a metric for how we measure that. So if I first start with ticks just out in nature, right? So this is ticks, Lone Star ticks. How long do they live versus nature? So had a master's student, Lindsay Bitter, who went out and actually, pardon me, this is an environmental study, so it's the wrong reference, but I have the other reference. She got this published. So she put ticks out in environmental captures. So she built these cans with a little bit of a support basket and mesh on the top, and then put the ticks inside of the bag and then put them out in the field site. And so the question was, how long does a tick live in the environment, right? So this is kind of the first stage of how long do they live outside? And her answer that she got, so let me walk you through this graph here. She would go out monthly and one for 4 months. So once a month, she would go out and pull out one of those canisters. She did 3D species of ticks. We're looking at Lone Stars here today. And then she would count what percent of the ticks were still alive at the end of each month. Now, the ones that were out for 2 months were out solid for 2 months. They didn't get brought in at the 1 month period. So they would bring in one can at 1 month, the second can at 2 months, the third can at 3 months, and the fourth can at 4 months. And the black solid line up here at the top was Lone Star ticks. So at the end of the experiment, she had, out of the 1,000 Lone Stars she put out there, 995 were still alive over the course of time, right? So the 250 that were out for 1 month were fine, 250 out for 2 months were fine, 250 out for 3 months, we lost a couple, and for 4 months we lost a couple. And so it really drives home Lone Stars can live out in the environment for a very long time. 4 months, they were still perfectly fine and happy. Questions about freezing, right? So we looked at that one winter where we said we had lots of cold, and maybe that did something for us, for the ticks. And so I had a student who was interested in this project. So he developed a similar study where he put ticks at different temperatures inside of a freezer and to see how long they would live. And so Duke did this work a few years ago looking at how they could survive sub-zero temperatures. We don't get all that cold here in Hampton Roads, but this past winter we got a little bit of cold. And so he came up with this little design where he put things in jars, and so he would always have sand or soil from the outside and then some leaf litter. And then he created what's called artificial snow. So he bought this artificial snow off Amazon and put it on top of them because his hypothesis was that snow would help keep them warmer. And then the ones that didn't have snow, so the ones without snow just had air above them. And then he put them in at -4 Celsius or 25 degrees Fahrenheit, so below freezing, or -18 Celsius or 0 Fahrenheit. So, and then he did both snow and no snow as he kept some alive on the counter just to make sure they didn't die in the setup. Okay. So he did this for all the different experiments. He did replicates and he put all those things in. And what he found was he would then pull one container every day. So again, similar to Lindsay's study, but as opposed to monthly, he just did it for a few days to test for survival. And so what he found was that all the ticks that were not in the freezer did fine, of course. Every tick at 0 Fahrenheit or -18 Celsius died after even one day. But we did have a good number of ticks that survived. About half of them survived without the snow, and about three or four survived with the snow at 25 degrees or negative, what was it, 5 degrees centigrade for 7 days. And so we learned then that this artificial snow actually desiccated the ticks, and so that's what helped dry out some of them to kill them. So I don't know if this is true about real snow, but we so rarely get real snow here in Hampton Roads, we didn't have that to test with. But it was very telling that ticks don't die from necessarily just the cold that we would get at ground level here. Our ground here in Hampton Roads certainly does not get below 25 Fahrenheit. The air temperature does occasionally, but not the ground, right? We also asked questions about, well, what about heat, right? So there was a study that the CDC did a number of years ago looking at black-legged ticks and whether your residential dryer could kill them. And their answer for the Ixodes scapularis was to take your clothes off when you got home or anything you were worried about and put it in the dryer for 6 minutes on high. And that seemed to do a great job for killing off Ixodes scapularis. And so they wanted us, and we worked with them to try and run the same experiment for Lone Stars to see what happens with Lone Star ticks. So we chucked our Lone Star ticks in the washer and dryer and did all the things they did. And what we studied, we found was it actually looks like it's going to take longer for Lone Stars than it did for black-legged ticks. And our best guess right now is 10 minutes on high rather than 6 minutes on high. So it takes almost twice as much desiccation or drying out or heat. If you wash your clothes first, it's going to take you over an hour of drying on high to kill the ticks with any consistency. And we're giving this as a guess right now because my statistics don't quite line up. So we're going to rerun a few of these this summer when we get our hands on some more adult ticks because the adult line here is a little bit extended out statistically. So we're waiting to see if we can't get a couple more data points to be able to understand what's happening and make a better precise guess and get that out. Had another student ask me about Lone Star ticks versus hurricanes, right? So when we had a hurricane that washed out Back Bay, as I was talking about, her question was, well, do ticks drown, right? I get to ask this a lot. People flush the ticks. I hear this often. And the question is, does it actually kill a tick to put it underwater? So we went out and we gathered water, salt water, brackish water, and fresh water. And then we put ticks in the bottom of them in these jars with the open tops. And so we could then set up this experiment to then pull out ticks every day, every other day for a month. So we set this up for 30 days to see how long ticks would survive underwater. And I figured that'd be plenty of time to get a good breadth curve. So we did these daily. You can see the daily time steps here now on the x-axis and survival rate again on the y-axis, same as we had for the environmental one. The blue line is fresh, the black line is brackish, and the red line is salt water. And you can see, well, it's at least 30 days. 30 days. Did I mention that part? 30 days underwater. We still had more than 80% of ticks alive in fresh water. Brackish and salt water, you started seeing a little bit of drop off after about 20 days. But still, you do not kill ticks off by putting them underwater for any considerable length of time. They have an amazing ability to survive underwater. And so the survival does not impact that washout that we saw at Back Bay, that one site on the ocean that I mentioned, isn't because it probably killed the ticks. It's probably driving out the host. So again, it's that complex understanding of how lone stars work, right? By the way, we continued that study because at that point, at 30 days, I was annoyed that the ticks hadn't all died, and we jokingly thought we were going to have to hand this off to like our children or grandchildren. So we continued the experiment, and it took almost 70 days for us to have all of the ticks die from being underwater. So it's an amazing amount of time that would be required for inundation. We don't get flooding here that often or that long. And so the answer was obviously, in this case, hurricanes would not be predicted to reduce tick populations. Cold would not be predicted. Environmental survival will not be predicted, right? So last thing we looked at for trying to understand the weather impacts was kind of a longer term view of it. And so we still have these hurricanes, we still have these flooding events, we have nor'easters, we have a lot of massive rain events. And I had an MPH student this past year who started saying, well, what if it's not a single event, but it's accumulation of events over time? And so, as I mentioned earlier, ticks live for multiple years. And so, at a multiple year accumulation period, we could then understand the multiple knock-on effects of all these ticks. And so, she looked at local flood events defined by the USGS, flood events for 2 years, flood events for 3 years, totals, averages, heavy rains, flooding and heavy rain. So all the data that you can get for what's happening in Hampton Roads area. And so she ran all of these things statistically through comparing to total Lone Star densities in our area. And what she found is that Lone Star tick count actually finally has some kind of relationship with the number of flood and heavy rain events in a 3 year total. The 1 and 2 year totals had no statistical relationship. It really takes that accumulation over 3 years of a lot of heavy rain and a lot of flooding events to have a decline in tick populations. And since we just found out that ticks don't drown, it probably takes that multiple year events to push the hosts out, which then pushes the ticks out over time. So the last thing I'm going to talk about, and then we'll open up for some discussion and questions, is people then start wanting to ask, is there any way to ever kill a tick? They are amazingly ubiquitous. They are amazingly hardy. We can't freeze them. I can't take my commercial dryer outside and dry the whole world. I can't drown them. They survive by themselves, right? And so we have come up with one more creative approach for how we are looking at tick control. We call these our little tick bots. So living on again the bay right by the ocean, it's not necessarily the best idea to just spray. A lot of places will spray acaricides, those insecticides, to your yard to try to kill ticks, and it's dangerous in the sense that it's not targeted to ticks only. It will impact all invertebrates. And so there's beneficial insects that can be in fact impacted and a whole bunch of other things. And here it will drain to the bay and can cause um, all kinds of death to invertebrates in the bay, which is bad. And so we kind of flipped this with the help of Dan Sonnenschein, flipped it on its head and a few engineers, where rather than spraying the acaricide or the insecticide on the ground, we spray it on a cloth or on the sides if they have wings, and then drag the cloth through the vegetation. And so rather than putting the chemical on the ground, we put the chemical on a cloth, dry it on there, it's secured on there, and then it's a permethrin. And when the tick touches the permethrin, it will die within about an hour. And so we can then run this up and down the field sites. We can run this along the edges. We can run this a lot of places. And then any tick that comes in contact with this would die. And so we wanted to prove this, right? So we went out to back to our Hoffler Creek Wildlife Preserve over in Portsmouth I talked about earlier, back in the years when it had a ton of ticks, not the years when it had few ticks. And we set up 3 transects and we tested them. We would go out the day before. We would collect every tick we could collect. We would paint it with fingernail polish. We would put it back. And then we would run our tick bot, and then we would look for ticks 1 hour, 4 hours the next day, and the next evening at those sites and compare recaptures of these painted ticks in treated and untreated areas. And what we quickly found was, and here, this is now those time frames I talked about. So at the setup point, as well as 1 hour after 4 hours, next morning, this one included in the next noon and the next afternoon. Red line is no treatment, and the blue line is treatment. And so what we found was we could, within an hour, drop the tick population to effectively zero for about 24 hours. So if you think about being able to make a space safe for ticks, it's great. Of course, the ticks come back. We don't get the ones that are in the leaf litter. We don't get the ones that are resting. We don't get the ones in the area. They're going to spread out. They're going to move. But it was nice to finally have something that we could do to at least like a Roomba, right, to vacuum the ticks up so that you can make a safe space. And so it did eventually essentially removes them to zero for 24 hours. They reintroduce themselves from those areas. So we are working with some engineers to improve this particular application and hopefully eventually commercialize it. So conclusions, right? Lone Star ticks are found in large numbers. They are very hard to kill. They are very different than other tick species in how you have to think about them and what you need to do to try to understand them from risk and to control. And then there's a lot of work that we need to do to look at control or reduction of human encountering so that we don't have these pathogens and illnesses that are associated with the Lone Stars. So, for example, my lab this summer, we're going to assess both tick-bot and residential yard treatments in a local homeowners association area, neighborhood that we can get in and try to look at more, even a granular level down to what do you do, how does that impact your neighbors, and where do you go from there? So stay tuned. I guess we got lots more work to do to understand what's happening and how these long-term data sets fit in and whether our numbers are going to go back up after a couple years of low numbers. So with that, I certainly have to acknowledge all the different funding sources that we've had for this over the many years of work and collections. Um, and undying gratitude to both the current Tick Team grad students and the previous 100 or so undergrads and grads that have been through the lab over the years, as well as my other faculty that have worked on this with me. And with that, I will open it up for questions.
Thank you so much, Holly. I think in all my years that seeing that graph of the underwater survival is my first startle response to a graph playing on a screen. So I don't know about everybody else, but so please, everyone, place your questions in that Q&A portion. We have plenty of time to answer questions, so type them in there. I'll just start from the top. So our first question is from Sarah Forsyth that there seems to be mixed data on the significance of turkeys as Lone Star tick hosts. Do you have an opinion as to whether they are significant, or could there be regional differences in turkeys as Lone Star tick hosts?
So we have certainly found that the sure the we did a little bit of work more with guinea fowl than turkeys, right? But we've looked at some of the larger birds, and so certainly they provide a really interesting ecological question where they do reduce tick populations, but probably equally because they're a host of those ticks, and they are eat them or consume them, right? Because so birds, guinea fowl, we did some, one of my PhD students, Alexis White, went out and actually looked at guinea fowl, and she did inclusions and exclusions and open areas and found that yes, guinea fowl reduced the tick populations, but she also did the horrific experience of sorting through all of the droppings of all of the guinea fowl under the coop for a couple weeks, and found engorged ticks. And so it was the catch-22 where they probably did lower the tick population by a good amount in that given year, but they're also feeding ticks. And so it's a question of like ecologically going forward in time, those one you miss one female and she'll lay 5,000 eggs and you're kind of right back where you started. And so that's a great open area of trying to understand that. It's almost like a predator prey switching where how those two interplay over time is still a really open question.
Very good. The next question is, in a dryer experiment, the data related to time in the dryer, did you check the dryer temperature?
Yes, yeah, we did, and I have all that data. When we publish it, we'll be able to share all of that. I didn't put it in here, of course, but yeah, we had a hygrothermometer, so we actually have the exact humidity and the exact temperature for all of the times, and the data were interesting. The my grad student Alexis White again actually was renting a house that had a old school electric dryer, the kind that make your zipper so hot that you burn yourself when you take your pants out of it. And hers would kill them within 2 or 3 minutes. And then I have the much more fancy modern gas dryer, and that's the one that takes the 10 to 15 minutes, it seems, to try to dry them. And so, yeah, we had the actual temperatures and tried to make sure all of that was parallel so that and that's where the high heat recommendation certainly came in.
Very good. A question about, did you test how well different tick repellents work on control of ticks? I'm going to assume that's on a person, but Mark, correct us if I'm wrong.
Yeah, so I think the best re so repellency wise, there's been a lot of work done. DEET works very well with repelling Ixodes scapularis. It does not work quite as well on lone stars. So black-legged ticks are much more easily persuaded to not feed on you. Lone stars, I swear they drink it for breakfast. They really, permethrin though is a neurotoxin, so it is not a repellent. It's actually an insecticide, so it will kill them. And so there's really not really much resistance to that, but it's not for your skin, right? Follow label instructions. It's good to put on your shoes or things like that. But um, other people have looked at cedar oils and some of the more natural event, and it kind of again depends on the older and more desperate a tick will get, the less the repellency will work. And so it's also a biological response of, I guess it's kind of like when you're really hungry, you'll eat anything. And I think ticks live by the same philosophy.
Very good. A question, when you drag a white sheet to catch ticks, do you lose many due to them falling off?
So we do have to be very cautious. So when we are particularly in the South, Lone Stars, no. Lone Stars will cling on to anything and they're running up that flag at you, right? But the other species of ticks that we're interested in, including the black-legged nymphs of the South, which are much more um, likely to fall off, as well as Ixodes kernsei, some of the non-human biters are a little more, I call them skittish. So when we're flagging, I teach my students we're very carefully leaving the flag on the ground. You watch it all the time. I'm sure we're losing some, absolutely. I'm not going to say we catch every one, but we are pretty aware and watching it all the time. You get a image in your mind pretty quickly to how to look for them. You're it's like where's Waldo? You eventually get to spot them, but yeah, and then even when you're trying to get them off the flag and into the jar, you'll drop one occasionally, or a student will lose their first one because it touches them and it causes panic, right? I mean, it's just a biological fact, so it's how it is. But um, Dragging, which you don't check as flag as often, I think is more apt to lose the southern ticks. But yeah, Lone Stars are Lone Stars. They're going to hold on.
Wow, I've got a lot more respect for Lone Star. Kind of scary. Um, it says, in a study with the different kinds of water, did you clarify, was there just the Lone Star tick that survived so many days? Did you test any other ticks? I guess is the question.
So the underwater experiment we also did Gulf Coast ticks, Amyloid maculatum, and it died much faster. So there was no extension of the maculatum study as much. That they definitely were they would live that 20 to 30 days and then be gone. So it definitely was much more Lone Stars were much hardier than all of our other local species.
So is this just the assumption that they're able to close their, well, spiracles, but they're how they breathe for that long?
The assumption they have is that they actually that last gas breath out, they put a bubble, right? They do they create a little bubble of air there, and then that has a gas exchange across that air barrier. So
From the
actually
water?
extract oxygen out of the water doing that. So yeah, nuts, huh?
Yeah, jeez. Um, it says, the question is, I've seen at least one tick testing company that is now including Borrelia lonestarii in their reports. Are you looking for Borrelia lonestarii, and how do you think it might be related to, is it
The
you
star
pronounce
eye.
it stari?
Yeah. That so for a long time people thought like the Borrelia lonestarii is the causative agent of this southern tick associated rash illness, but we haven't like it's so inconsistent in the findings. And some of it is that Borrelia are really hard to work with in that they're intercellular and they're endothelial cells, they're not in your bloodstream, and you don't often have the tick to test for it. So that was the theory for quite some time, and then I've seen people back off from that, and I am not a microbiologist, so that's pretty much the extent of what I know is it's really, really hard and they don't know, but I think um same with a lot of the other rickettsias, we don't know whether they're human pathogens or not, but they're exposing us and probably have some kind of immunological reaction. So um those are great questions that we need people to work on.
Yes, very good. Next question is, how do you collect the larvae from the flags, and how do you store them from the field to the lab?
So the larvae off the flags we collect with blue masking tape, the paint tape that you use at the store, right? That's our best collection method. I swear we should be sponsored by Home Depot sometimes or Lowe's or some hardware store, but the blue painter's tape takes the ticks off very nicely, and then you can roll it up and put it in tubes. And so we have them on with little snap top vials that keep them secured in the Ziploc bags and then take them back to the freezer. And then since we now know freezing will kill them after a week, we put them in there for that week and then we can pull them back out and safely sort them.
Very good. Another question, what tweezers or forceps work best? What do you prefer when working with ticks?
Yeah, so we have when I do field work, I use very like child-friendly blunt-nosed ones because they tend to walk off otherwise, and specifically because we take those larvae and nymphs with tape rather than by trying to pick them up with forceps in the field because you're out there, it is hot, and there are thousands of them and you are sweating. So that in the field, we definitely use very blunt forceps, and then back to the lab, we get the nicer ones out in the lab, but pointy tweezers. But yeah, pointy tweezers in the field is a bad idea, in my experience.
Yes. Um, Next question is your thoughts on the impact of soil composition, clays, minerals, silica, pH on tick survival.
So we started going down that understanding. We do have one particular tick species that is strictly limited to the coastal plain and it doesn't extend into the Piedmont very far. And our theory was that there's got to be some geological differences. Um, and what was fascinating to me was like, I mean, again, my background, full disclosure, I am a mathematician, and I'm like, there's at some point I run out of. We tried some experiments looking at pH. We tried some experiments, but we also have to then have the understanding of that, at least some of the soil compositions that can change um, over time, is how 3 years of that data gets accumulated together, right? It's definitely not as simple as a single snapshot. Some of the soils don't change, right? So soil composition probably doesn't change a whole lot, but they live up in that top layer, which changes considerably with fall and then leaf litter and the breakdown, and that situation changes quite a bit. So it's pretty amazing how much variation there is at their level, and then rainfall and everything else. But no, absolutely. If someone wants to come work on that, we can find a good PhD project right there.
Very good. Another kind of materials and methods, do you use a square meter drag or one with strips?
So for the regular flagging, we use a square meter. The strip one we use when we're doing any kind of like the tick bot has that gets down in between. Um, but for us, the square meter is what we started with, and so at this point, 350,000 ticks in, I'm not changing it. So I don't. We get plenty of ticks, so I think as long as yeah, we've gone with a solid.
Okay, our next question says, I have alpha-gal. Physicians still aren't well versed on it. From the science perspective, do you believe this, which I assume means Lone Star ticks, is a one way we humans get alpha-gal? And just in parentheses, some victims think it's could be bioterrorism.
Yeah, so alpha-gal itself, I think it's we've known about that reaction since a lot of the transplant work, right? So if you look back through time, anytime we started trying to do transplantation with pork products, we started seeing alpha-gal come up, right? It is a known biological reaction, probably related to fighting off bacteria way back in the day evolutionarily. Um, there are certainly many routes of exposure, not just Lone Stars. We are now understanding that Ixodes scapularis. There are a number of other ticks throughout the world that can transmit, that can trigger that. It's not transmit, it's triggering the allergy. There's actually evidence that there are some helminths, so some worms that also do this. And so there's probably something more complex evolutionarily in this interface that we don't yet fully understand, but it's certainly, um, given its long history and how long it's been around, it's a natural process, in my opinion.
Thank you. Um, Best forceps are tool to use for tick removal from humans and pets.
So originally, the CDC was all about making sure everyone only pointy tweezers, and I think it's more. Um, we give out tick spoons, these little spoons that have a slot in them that help pull it out. Use your fingernails. The real key is to get it out of your skin as quickly as possible. People worry about leaving the head in, but ticks don't really have heads. They have a mouth part that's probably going to be like a splinter as it grows out. Um, and so the best would be a pointy tweezers straight against your skin and pull straight up. That's the ideal world. But if you are out in the field, don't wait till you come home to find pointy tweezers, right? We like that tick spoon. I've seen a new little ID card thing that little has a little slot on it, anything like that. Or if you have good fingernails, you can probably get the body out. I do recommend keeping the tick, right? It helps identify what pathogens you could have been exposed to if we know what tick it is. Um, and there are places that test ticks. We test ticks. That's not necessarily medical information at this point, but sometimes it can be useful information.
I know with Lyme disease, we kind of usually say 24 to 48 hours of feeding. Is this similar with the Lone Star tick vector diseases?
So it's a highly variation, right? So the 24 to 48 is an estimate for Lyme. I think there are ways it can be faster, but the rickettsial agents we know are in the 4 to 8 hour range.
Mm.
And then what terrifies me, of course, are things like the viruses, which are 15 minutes. And alpha-gal saliva triggering reaction is probably closer to that 30-minute range. So it's early in the saliva exposure. So things that are already in the salivary glands as opposed to those that have to transit to the salivary glands during feeding. Um, but
Very interesting. Well, thank you so much. I'm going to give everyone the last few minutes to type in any other last minute questions as I just let everyone know that our next Pests in Progress webinar will be Wednesday, May 20th at 12 p.m. Central, 1 p.m. Eastern. We're going to do RNAi and varroa mites, a new technology for managing an old pest of honeybees with Matt O'Neill and Jared Perez from Iowa State University. I know we've dropped in the chat how to subscribe to our newsletter. I just wanted to give everyone a heads up who's attending this one on January 20th. We have Eden Stewart from the University of Missouri. She's a dietitian and kind of addressing the alpha-gal syndrome. So that one might be of
mhm.
interest to this audience as well. And so with that time, we got a couple other questions. Your thoughts on trying to remove ticks with fire, oil, or any way to make it detach from your skin instead of tweezers?
Yeah, so there's always these great videos online of ticks backing out or, irritate the tick. I mean, these suckers are designed to be in there for, well, the immatures, the larvae, nymphs, and the females are designed to lock themselves in for the next, 3 to 21 days. And so very little, it takes them about an hour to get settled in, and it'll take them about an hour to get unsettled. And so anything you do to irritate them to get out of there probably isn't going to be any more irritating than an animal's sharp, pointy teeth chewing on it. So you're not going to encourage it. A lot of the ones I've seen, if the videos, if you look at them carefully, it's a male tick, which take very small blood meals and just go mate. And so they're more likely to be, or it's early in the process. It's not certainly fire. I would never recommend, right? Smothering them, as you just learned, they can hold their breath for 71 days. Or 70 days. They're not gonna die from suffocation. And so it's not removal by physical removal is your best bet, and you're not. People worry about squeezing the things into the ticks and they don't regurgitate. It doesn't really work that way, but if you get that close to your skin and pull straight up.
Okay. Um, Question on, I guess, how do you balance you and your lab balance personal protection when collecting ticks versus sometimes you're collecting them on your clothes as well.
Yeah, yeah, the rule in the lab is don't be a data point, right? Yeah, we have biosafety protocols that we have to follow, right? But the easiest thing we do is we wear rubber boots up to our knees. We're also in swamps, and so that also helps with that. But the rubber boots are better than laces because larvae can go through laces for lone stars at least. And so during high tick season, we'll duct tape our pants to our boots, and then we'll tuck our shirt in. And it is again a lot of mutual grooming. So we take care of each other, check when we get back to the car. Um, it's statistically, I tell my students at this point still more dangerous to drive to the field site than it is to be at the field site, even collecting ticks actively, but definitely try to make sure we pay attention and tick check, tick check, tick check. So it definitely is it's a way of life.
Yeah, that's a good point. And with some of those short bite times, I mean, I think we usually say, oh, when you get home, check. But I mean, I think people should just be remembered
Yeah.
to be kind of persistently checking when you're
Yeah.
outdoors in tick areas.
Yeah, and I do encourage my students to treat those boots with permethrin, so that way the tick crawls across it, they will be less likely to be alive still when they try to start biting. So that helps. But again, that's a personal choice on chemical use.
Yes. Um, we've had a lot of questions just on when they're going to get a copy of your talk. So just to reassure everyone, we will create the transcript and the closed captioning for it. And so if you're registered, you'll definitely get an email when it's available, and it will be on YouTube, so you'll be able to watch it at any time. I don't want to promise how fast. Usually within the week, probably later this week, but you'll get that email pretty quickly and be able to share the information. So thank you again so much.
Thank
This was
you.
really informative. You got lots of, if you haven't been reading, there was lots of great comments from everyone. So definitely, serious problems, serious diseases, and it's great to learn a lot more about it. So we really appreciate it.
Yep, no, I appreciate it and I appreciate the opportunity, and the more we can get the information out, the better we are. So,
Definitely. So have a great day,
all
everyone,
right,
and we'll
thank
see you later.
you.